this is Jynessa, and I will be talking about DNA cloning. It's first important to discuss what exactly DNA is. DNA is a polymer composed of two polynucleotides that carry genetic information of an organism. So, what exactly is DNA cloning? DNA cloning is the process of making multiple identical copies of a target strand of DNA. DNA cloning is done in 4 major steps. First, the target DNA strand has to be isolated. Then that DNA strand is inserted into a plasmid found in a bacterial cell. That strand or rDNA connected to the plasmid is then transferred into that bacterial cell. The rDNA or recombinant clone is then detected and purified within the cell. How exactly is DNA cloning used? DNA cloning is used in many ways, such as biopharmaceuticals, gene therapy, and even gene analysis. In biopharmaceuticals, DNA cloning is being used to make human proteins that are used in medicine like insulin. In gene therapy, it provides patients with a gene that the patient is missing due to specific genetic disorders. Lastly, in gene analysis, DNA cloning allows researchers to observe and understand the functions of some genes. Let's focus a little more on how DNA cloning works in biopharmaceuticals. One way DNA cloning is used in biopharmaceuticals is by the making of insulin. The process of DNA cloning starts with the plasmid of a chosen adult bacterial cell and a cut strand of the target cell. In the case of making insulin, the bacteria, E. coli, would be used. The DNA of interest is broken or fragmented in order to provide the target DNA strand, or gene. The plasmid of the chosen bacterial cell, E. coli, is cut with restriction enzymes in order to make space for the isolated target gene. This DNA fragment is then connected to the circular loop of bacterial plasmid using a sticky protein called DNA ligase to stitch the pieces together. With all of that said, let's take a closer look at the diagram. On the left-hand side, we have the DNA sequence, and we also have a plasmid that comes from a bacterial cell. You can see that sections of it are cut off on both the plasmid and DNA sequence. In the second picture, we have the DNA of interest in the plasmid, and you can see how it connects. And the last picture is the plasmid with the DNA connected by DNA ligase, and that creates what is to be cloned. To look a little closer, in orange we have the DNA sequence again, and in the green is the plasmid loop of bacteria DNA from that bacterial cell. In this picture here, we have the DNA connected to that plasmid through DNA ligase, and that is what will be cloned. Once the DNA strand is connected to the plasmid, it makes what is known as the recombinant DNA. The bacterial cell is prepped by being given a shock that allows the bacterial cell to take in the new gene. The recombinant DNA is then placed back into the E. coli bacterial cell. On the left-hand side, we have a bunch of bacterial cells that go through different levels of shocks. On the right hand is the surviving bacteria, which is the one that survived from being shocked. That bacteria will be able to take in the plasmid. This bacterial cell is an adult cell, which means it will reproduce and pass on the recombinant DNA plasmid to its offspring, as seen in the image. That is how the recombinant cell is then copied multiple times, making the clones. Once the DNA is replicated through the E. coli, it must be purified from other proteins and materials within the bacteria cell using biochemical techniques. Some examples of biochemical techniques involved in purifying the recombinant DNA plasmid includes chromatography techniques with ultra-difiltration systems and sensitive analytical procedures that help to detect the impurities. The final results in case of DNA cloning in E. coli are the protein insulin, which is commonly used in the medical field for diabetes. Today, in DNA cloning, animals are being cloned. In one case, pigs are being cloned in hopes to make organs more suitable for humans for organ transplants. Monkeys are also being cloned for more research. A U.S. fertility doctor has claimed to have started a process to clone humans. Continued research on DNA cloning is being done to help cure diseases. And also, ethics about DNA cloning are currently being questioned. That is all for this video. Thank you for watching, and I hope you enjoyed.